It is a real pleasure to take part in this session with such a fantastic roster of speakers. And I want to thank the organizers for the chance to address such a diverse audience and speak about our company, Purissima. Special thanks to Keith and all Life Science Angels for all your continuous and tremendous support of Purissima over the years. And what I would like to do today is I want to talk about our company Purissima and about medicines. Now, medicines are essential for our quality of life, and yet we lack safe and effective medicines for many of our most pressing diseases and conditions, such as, for example, neurological or metabolic disorders. Now, many of these limitations are very closely tied to how we get our medicines. What I'm going to show you today is how Purissima deploys modern toolkits based on synthetic biology approaches and genetic engineering to solve some of those problems and ultimately to map meaningful segues into the future. Rob and I worked together for over 10 years since we have first met at one of the first synthetic biology companies based here in San Francisco Bay Area by the name of Solazyme, now known as Corbion. Since then, we moved on and successfully combined technology and business experience to launch our darling Purissima. We formed the company in 2017 to really think how we could address some of the most fundamental problems that we all face as humans at some point in our lives, and related to pain, stress, anxiety, and mood, and as potentially very often as a result, our personal appearance and overall well-being. And of course, how we can utilize newly emerging Synbio approaches, and how we can build a successful business around it. Now, existing mechanisms of or many societal lubricants to cope with pain, stress, or mood-related problems represent large markets. But since broader legalization of cannabis and cannabis-related products in the United States a few years back, and probably most notably in California in 2016 with Prop 64, and after introducing the Federal Hemp Farm Bill of 2018, there is a clear and serious erosion in these existing mechanisms. People started to use cannabis products instead. And not only cannabis-related products threaten existing markets, but their introduction to the market has additionally precipitated a really broad and ongoing broader national discussion on how effective is our traditional health care system as a whole, and are we missing out on simpler, safer, and cheaper alternatives, and most notably natural medicines and remedies derived from plants, let's say from traditional medicine plants, and all compared to existing medicine and pharmaceutical drugs. Let's take a look at one of such examples. Opioids are essential medicines that are used to treat moderate or severe pain, and they're widely used across the globe in palliative care and in many, and for many of us in our daily lives. Yet, World Health Organization has estimated that over 5 billion people on the planet do not have sufficient access to these medicines, and most notably because of higher costs. In this particular example, we also have the case where there's abuse of these compounds, and in the United States, we are facing an epidemic that is also a result of a healthcare system that has prioritized profits. So how do reconcile the need for therapeutic benefits, challenges with supply access, and the safety of medicines themselves? And can we think about additional ways of developing safe and efficient medicines? What's notable in that specific example is that two of the opioids, codeine and morphine, are natural products from plants, whereas the other ones are synthetic chemistry modifications to improve that were created to improve therapeutic potency or bioavailability, thus combining natural product with biochemical synthetic synthesis. In fact, of the marketed pharmaceutical therapeutics, approximately 1/2 can be traced to natural products or are derived from natural products, whereas only about 1/4 are purely synthetic in origin. However, the supply of natural products is challenging and cannot fill the ever-growing discovery pipelines for most pharmaceutical companies. Combinatorial chemistry techniques have been successful and revolutionary in developing new drugs and in optimizing structures of many recently approved compounds. Yet the output in small molecule discovery pipelines using pure chemistry shows really diminishing returns, whereas drugs based on natural products with well-defined chirality have been marketed year after year after year. So we decided to focus on developing new approaches to combine natural products, reservoir, and chemistry in making additional drug discovery streams. Now, uncovering medicines from nature, and more specifically from plants or medicinal plants, has inherent limitations as well as the first. And the first one probably is that these molecules are typically made in vanishingly small amounts, very often in specialized cell types and organs, such as these tiny trichomes on the leaf or stem surface, shown on top left of the picture, or secretion cells in opium poppy and resin ducts in conifers, or in specialized organ formation, such as nodules in the roots of legumes plants, shown on the lower right photo. Compounds often originate from rare medicinal plants that grow quite slowly, sometimes a year long, and in very particular environments. And so sourcing enough of these molecules from those rare plants could be very challenging, and in many cases, very difficult to do in an environmentally friendly way. And often we wind up over-harvesting these plants and then losing irreplaceable diversity from the natural world. The second challenge is that these molecules then have to be improved, as nature is not making them as medicines for humans. So as a result, these various challenges can lead to decades of research and development that are required to take a molecule and bring it to the market as a medicine, and these challenges essentially create barriers in developing new and better medicines. The question then becomes, how do we address these challenges? How do we develop technology to enable the production of truly efficient, active, and safe ingredients manufactured in a sustainable way and be widely accessible? And one of the ways to bring change to the system would be to rebuild chains that supply our medicines. And we started this journey with the cell, plant-like cell, and we have chosen a beautiful microalgae cell. And so we placed the cell in a very familiar standard industrial fermentation vessel, similar to what is used for brewing beer, and asked if we could provide it with genetic instructions from plants to make specific complex molecules that would still maintain their properties and essentially replace plants in making these compounds with the best possible proxy environment. In this way, microalgae could serve as a manufacturing platform with rapid cell doubling time, and also they have the ability to grow on inexpensive nutrients and at large scale that would make the whole process very inexpensive. The production cycle as well is days, not months as for many plants, and the process itself is intrinsically sustainable and climate independent, and does not include many pesticides or land or large energy needs as needed for typical agricultural production. So microalgae are naturally optimized to produce complex lipids. So we have tested this back at Solazyme days probably over 10 years ago with engineering of triglyceride oils. And shown here are a few public examples of making tailored triglyceride oils that are composed of various fatty acids and matching the properties and composition of some existing commodity oils. And this was done with engineered microalgae that could be scaled and manufactured at over 500,000 liter fermentation tanks, which was a very tremendous achievement at that time. So since then, the synthetic biology moved on, and most notably in terms of capturing genetic information from thousands of living organisms, including plants and many medicinal plants, all through large-scale DNA sequencing efforts and genomics tool development. This vast information, in essence, unlocked the blueprint of biosynthetic processes that occur in nature and that we can utilize in order to recode in our microalgae cells and to produce complex natural ingredients, novel and interesting compounds and intermediates that could be used in drug development. As I have mentioned, microalgae are prolific natural producers of complex hydrocarbon lipids and as well as many secondary metabolites. In fact, we now looked at three major classes of these molecules that comprise about 75% of natural secondary metabolites, and this list obviously growing every day. Many of these compounds have direct and diverse practical applications that we are interested in, and many of these are used as drugs or remedies today for various medical treatments. The beauty of our technology is that we have instructed microalgae to produce compounds in every of each of these classes. And as with triglyceride oils that belong to acetate polymalonate group of class of compounds, we have now accomplished the production of multiple compounds or their intermediate precursors, such as complex prenulated lipids, vitamins, alkaloids, and polyketides. Interestingly, and looking at chemical intersection of these chemicals, we realized that bringing the specialized enzymes or specific biosynthetic pathways alone, or possibly in combinations that naturally don't occur and into our algae, opens up an extremely captivating opportunity to enhance the natural compound diversity through engineering truly hybrid chemical scaffolds or their modifications. And this would be so-called natural, unnatural compounds, as they are assembled with natural chiral properties in the living cells, but they don't naturally exist in nature. So now let's look in a little bit closer at what this manufacturing platform actually looks like. And I would like to dive in using cannabinoids as a class and as an example. Cannabinoids have a potential to become indispensable in our daily lives, and as they have very diverse applications, and perhaps outside of the psychotropic properties of THC. Those properties include, most notably, mitigation of pain and anxiety. They help with stress relief. They serve as sleep aid, and they're natural antibiotics. Now, there are 4 approved drugs with anti-epileptic drug Epidiolex, which is based on cannabidiol or CBD, most recently developed by GW Pharma to treat epilepsy-related seizures associated with Lennox-Gastaut syndrome, Dravet syndrome, and tuberous sclerosis complex in kids. Cannabinoids can potentially be developed into drugs to treat many neurological disorders, as shown on this diagram here. And since cannabis legalization, the new wave of research studies would certainly yield very interesting results in the near future. And just as an example, there are over 500 clinical trials that are currently registered with NIH. And this obviously makes it a very exciting time for cannabinoid research. Okay, as shown here for cannabidiol, cannabinoids are complex molecules with phenyl ring serving as the core element, and prenyl and alkyl groups being more variable. Combinations of these 3 elements derived from separate biosynthetic pathways could potentially create a large number of interesting compounds. This, for example, major classes shown here, demonstrating variability in alkyl chain or phenyl ring. So the question then becomes: can we replicate cannabinoid production in microalgae and can we make novel cannabinoid structures? Well, when we looked at the cannabis plant genetic makeup to understand biosynthetic pathway, we were surprised to discover that genes encoding specific biosynthetic steps, such as the gene encoding CBD synthase catalyst shown here, exist in families with many members that are slightly differ from each other. And if you look, for example, if you look at the sequence alignment shown here, also the genetic makeup among, for example, different cannabis cultivars, such as a marijuana plant and the hemp cultivar, are not identical either. Now, the nature incorporated a lot of redundancy in this biosynthetic pathway, and different cultivars evolved to make different cannabinoid compositions, but that clearly creates a lot of challenges for classical breeding, as it's really unclear which of the gene variants encodes the correct or the best catalyst. Now, our approach offers a clear advantage, as we can quickly bring these genes in microalgae one by one or in combinations, and then subsequently identify the correct ones. And microalgae cells generally help to untangle these complex biosynthetic networks because they have a similar architecture as plant cells. The cannabinoid biosynthesis in cannabis sativa is a sort of very choreographed process taking place between different compartments in the cell, with initial precursor molecules made in the cytosol, and then subsequent key catalytic reactions taking place in the plastid, or known as chloroplast, or in endoplasmic reticulum, the two compartments that evolved by nature to make the vast majority of secondary metabolites in plants. And this is how key major cannabinoid molecules are made, such as CBD, THC, and others. And when successful, the nature tends to recycle biosynthetic modules over and over again. And in our case, there is an almost identical biosynthetic process that is described in a rare plant, rhododendron dauricum, that makes same classical cannabinoid chemical scaffolds, but different side groups. Now, dauricaminic acid, which is one of the major products of this biosynthetic process, is a potent anti-HIV drug candidate, but it could not be sourced sustainably, as rhododendron dauricum grows in Hokkaido in Japan and is designated as a protected species. And this is one of the challenges with medicinal plants that we talked earlier. So what we did, we replicated both biosynthetic processes in microalgae and generated not just the compounds shown here, but also some additional compounds that exist as traces in these plants. And then we ask the question, can we combine parts of one of these pathways with parts from another pathway, combining cannabis steps with rhododendron steps, and create truly hybrid cannabinoid structures that don't exist in nature? And to our surprise, or maybe not, the answer is yes. We can combine parts of cannabinoid molecules in a truly combinatorial way and allow biosynthesis of novel natural and unnatural compounds, starting with different prenyl and alkyl groups that distinguish these two types of cannabinoid outcomes. So we integrated this purely genetic approaches with novel computational tools on the next level, tools that enable further improvement in the biosynthetic process by improving the actual catalysts or changing potentially their specificity. For example, we look at the really at billions of conformations here to calculate the best fit between two important precursors. And subsequently, we evolved the catalyst to support the desired chemistry. And while scaling up fermentation to generate first cannabinoid products and introduce them to the market space, we have created the first library of very unique compounds that include novel and natural structure that show promise in the clinical studies. So we created a library of over 70 different compounds today, and this list is growing essentially on the weekly basis. With this first library of compounds, we have initiated preclinical studies with some of those molecules targeting both consumer and pharma applications. On the consumer front, we have interesting data for various skin conditions and overall wellness responses, such as those related to pain, sleep, or stress. On pharma front, we have very interesting initial responses related to pain management in some autoimmune disorders, as well as good leads related to neurological conditions and even tuberculosis. So the importance of this is that with our technology, it's not that we developed an improved manufacturing platform by fusing synthetic biology with age-old fermentation and to deal, let's say, with medicine shortage, but it's also the ability to go beyond what we find in nature and ultimately to create better medicines with improved therapeutic activities and reduced negative side effects, as for example, needed in pain management. And as a company, we are looking at developing tailored wellness products to ensure the reliable and repeatable customer experience, because this is exactly what our fermentation platform and just-in-time manufacturing could provide. So now coming back to the very beginning, when I emphasized or talked a little bit about our high-level long-term mission, and with the progress that we have so far, we are really looking forward and eager to address some of the most underserved conditions, and particularly those related to women's health and wellness. And of course, more generally, we would like to keep on developing truly novel or interesting solution for CNS and mental health segments. So as the company, we have developed a really powerful bioengineering platform with unique ability to make novel compounds protected by strong IP. The company has two major strategic partners, and we expect to generate product sales already sometime this summer. Finally, I'd like to thank you for your time and welcome you to join us on this existing journey. Help us in all possible ways by potentially collaborating, or perhaps we can think about some interesting clinical models, or maybe it's just a computation. And obviously, as any startup, we are always looking and searching for money. But we are on a very interesting trajectory, and I'm really excited about our future and about what we can and what value we can bring to the people. And so thank you, and thank you for your time again.